Bismillahirrahmanirrahim. We continue with the June 24 question paper 1-1, and we have question 20 to question 40 in this video. We're starting with question number 20. Which row is correct for the start of anaphase of mitosis? Anaphase is one in which they move apart. So they are separated sister chromatids. Now then you've got to figure it out further. DNA is associated with histone. Please remember, mitosis is only to do with eukaryotes. It never happens in prokaryotes. Bacteria only divide by B for bacteria, B for binary fission. So bacteria is not mitosis. So there'll always be histone. So always. And the cell membrane is always going to be intact. Why should the cell membrane be destroyed? So question number 20, that's why the answer is C. Cell membrane is never broken apart. I mean, this is to confuse you all because the nuclear membrane does break apart and then of course is reformed. Then coming to question number 21. The diagram shows the chemical structures of 4 bases. Now the 4 bases, as you know, are what? Adenine and guanine. Adenine and guanine are purines. And purines have a double ring. This is just a quick revision. Now what is left of the other three? There's cytosine, uracil, and thymine. These are pyrimidines. These are single ring. You have to make some mnemonic to remember these. Now, which diagram shows thymine and cytosine? So thymine and cytosine are what? Thymine and cytosine are single ring. Now this is the only one which is single ring, and this is the only one which is single ring. So which diagram shows thymine and cytosine? So it is 2 and 3. But you have to remember this, that adenine and guanine are purines and they have a double ring. There's a U in purine and the U in double ring. Cytosine and thymine and uracil are pyrimidines. Now please pause the video here and have a look at it. That's why the answer is B. It says the diagram shows the nucleotide sequence of a small section of the transcribed strand of a gene. So this is the DNA, double helix. And this one is a transcribed strand. So this is the mRNA being made. So they are telling you that this is the transcribed strand. So the DNA has CGG, GCC. So you've got to figure this out. What is it going to be? So this is how I figured it out. I've shown it to you with different colors. And question number 22. The answer is B, and please look at it very carefully and have a look at it. So the DNA, you should have made the mRNA because it says the amino acid short-coded by 10 mRNA codons. So these were the mRNA codon and this is the amino acid. So you have to figure this out. What does the process of translation require? What does the process of translation require? Where is transcription is DNA to mRNA. Translation is mRNA to protein. So it's very easy to figure this out. The answer was D. Translation requires an mRNA, ribosomes, amino acids, and tRNA. Question number 24. The electron micrograph shows a longitudinal section of part of the stem of a plant. What is the name of the structure labeled X? How are you going to figure this out? So how are you going to figure this out? What is X? Now I can see here this is the sieve tubes. You can see this here. Those are the sieve tubes, sieve plates. So this is the thing which was going to give you the clue of it. And this would be the phloem sieve tube element. Then question number 25, which substance makes xylem vessels impermeable to water? Please remember cellulose cell walls are fully permeable. Suberin is not found in the xylem vessels and collagen is not found in the xylem. So the answer is B, which is lignin. 25. The answer is B, lignin. Question number 26, direct question. The following tissues carry an electrical impulse during the cardiac cycle: atrioventricular node, muscle wall of the atria, Purkinje tissue, sinoatrial node. In which order does the electrical impulse travel during the cardiac cycle? Either you know it or you don't know it. It has to start with 4, which is the sinoatrial node, and the answer is 3. Why? Sinoatrial node, then muscle wall of the atria, then atrioventricular node, and then the final is the Purkinje tissue. Then coming to question number 27, the graph shows the changes in the volume of the ventricles during a single cardiac cycle. So volume of the ventricles. Volume of the ventricles in arbitrary units, this is the y-axis and this is the time. Which row is correct for the atrioventricular valve at P and for the seminar valve at R? Now, atrioventricular valve at P, now when the volume, ventricular systole volume of the ventricles has risen, so and then it remains high and then it starts to decrease, so ventricular systole is starting, so atrioventricular wall must be closed. It's either this or this. Now, in semilunar valve at R, what is happening? The volume, so ventricular systole has taken place, the ventricles are now empty, and diastole is going to start, which means the relaxation is going to start. So, semilunar valves at R will also be closed. So, that is why the answer is A, because they'll both be closed. You have to look at the volume of the ventricles. You have to look at this is atrial systole, then this is ventricular systole, and this is diastole. Diastole means relaxation, systole means contraction. So when the ventricles are contracting, when the ventricles are relaxing, or when the atria are contracting. Question number 28. The graph shows how changes in oxygen concentration affect the percentage oxygen saturation of human hemoglobin and cat hemoglobin under normal physiological conditions. The partial pressure of carbon dioxide was kept constant at 5 kilopascals, and the temperature was kept constant at 37 degrees Celsius. So you can see this is the human graph and this is the cat graph. Now, which conclusion is supported by the graph? Now, I always tell you is you draw a graph here, you draw a line here, and you look at it. So, percentage saturation of hemoglobin, say this was about 30%. The human hemoglobin was about 60%. You must give figures on the graph. Partial pressure can be 1, 2, 3, 4, 5, 6, something like this. So, at say 3 partial pressure of oxygen, cat was only 30% saturated and human beings were 60% saturated. Right? Now, this helps you to make certain conclusions about the graph. Which conclusion is supported by the graph? Now, the answer is C. Why is the answer C? At low partial pressure of oxygen, oxygen is released more easily from cat hemoglobin than from human hemoglobin. So at 3 partial pressure, you can say the cat hemoglobin was only 30%, while the human hemoglobin was 60% saturated. It says which conclusion is supported by the graph. So you've got to say this to yourself. Does the graph tell me this? Now this question, the graph is not telling you this. The shift in the oxygen resources caused by the Bohr effect is larger. Forget Bohr effect is when the CO2 changes. There's no Bohr effect here. Here they told you in the question that the partial pressure of carbon dioxide was kept constant at 5 kilopascals. Bohr effect only comes into operation when the carbon dioxide concentration is changed. So this is how you read the question and this is how you understand the graph question. Then coming on to question number 29, which statement is correct about how oxygen combines with hemoglobin? So question number 29, the answer is B. One oxygen molecule can combine with each heme group of the hemoglobin molecule. So that's why it's 8 atoms. Now, combining all 4 oxygen molecules with hemoglobin does not affect the shape of this is wrong. It does affect the shape. The first one affects when it attaches, changes the shape, and then it facilitates the other 3 binding. The first oxygen molecule combined does not affect the shape. Again, this is wrong. The third oxygen molecule to combine with, he makes it more difficult. No, it's the other way around, it makes it more easier. Then question number 30, how are alveoli adapted to their function? The answer is A, they contain squamous epithelium for a short diffusion distance. Alveoli, sorry, do not contain any goblet cells. They contain ciliated epithelium, that's in the trachea. They contain cartilage, that's in the trachea. Question number 31. The diagram shows oxygen diffusing from the space inside an alveolus into the blood through the gaseous exchange surface. Now, I always tell you ways how to handle these questions. So always put some figures on it. It says thickness of the alveolar wall and capillary endothelium. That is Y. W is the oxygen concentration in the blood, and X is the oxygen concentration in the alveolar. So say this is 100 and inside it's 50. Now it says what would increase the rate of diffusion of oxygen from the alveolus. So from the alveolus into the blood. Now what should increase? Now if we increase X, the oxygen concentration, make this 120. So this would increase the gradient, the steepness of the gradient of diffusion. That's one of the points written in your books about what would increase the rate of diffusion. So if we increase X, it will become 120. This would, of course, increase. Y decrease? Y means the width of this. The thickness of the alveolar wall and the capillary therapy of that would decrease. Yes, it would help. And W can keep the same. This can remain the same, the oxygen concentration in the blood. But if we make this from 100, we make this to 120, so it becomes, the gradient becomes steeper. So that is why the answer is D to question number 31. Question number 32, which tissues are present in a bronchus? Cartilage, ciliated epithelium, and smooth muscle, all three of them. So question number 32, it's A. Question number 33, steep concentration gradients must be maintained. Steep. First understand the question. Steep must be maintained for efficient gas exchange to occur. Why should alveoli always have more oxygen? Blood will always have less oxygen which is returning to the lungs. Pulmonary artery bringing deoxygenated blood to the lungs. Which row correctly describes how steep gradients are maintained? Elastic fiber in the walls of the air recoil when breathing out. Yeah, because that is going to refresh the air. The oxygen is vented in. Now the alveoli has less oxygen. So, but when you exhale, the elastic fibers in the walls of the alveoli recoil when breathing out. That's going to help because this low oxygen in the exhaled air is moved out and fresh air moves in. Continuous supply of deoxygenated blood by the pulmonary artery. So this is always less oxygen. It will keep on coming less oxygen and then getting saturated with oxygen and then moving ahead. So 33 answer is A. Then we come to question number 34. Now, a scientist investigated the effect of an antibiotic on the treatment of cholera. 320 people with cholera were divided into two groups. One group was treated with antibiotic, while the other group was not given the antibiotic. Both groups were given fluids containing sugars and mineral salts, oral rehydration therapy. The scientists recorded the number of days that each person had diarrhea. The table shows the results. Treatment antibiotic and oral rehydration was 3.2 days. That meantime the person had diarrhea. Oral rehydration therapy, no antibiotics, was 5.3. So what is the percentage decrease in the meantime? So 5.3 minus 3.2 is equal to 2.1. Divided by what? You will divide it by 5.3 and multiply it by 100. And the answer is this, it's A, 39.6%. You see, percentage decrease means change over the original. The original oral rehydration therapy was 5.3, so you take the difference and divide it by 5.3, and then you get the answer. Then coming to question number 35. How does antibiotic penicillin affect the metabolism of a bacterial cell? Now the answer is B. Penicillin inhibits the formation of cross-links in the cell wall between peptidoglycan molecules. And that's a very factual thing. You must know it. Why penicillin does how does it work on it? It inhibits the cross-links in the cell wall between the peptidoglycan molecules. Question number 36. Scientists studied the multi-drug resistant bacteria infection in children caused by one type of bacteria. The percentage of multi-drug resistance rose from 0.2 to 1.5%. So 1.5 minus 0.2, 1.3. You divided it by 0.2 and then into 100, and that's D, 650%. Then coming to question number 37. Whooping cough is a highly infectious disease of the gas exchange system caused by the bacterium Bordetella pertussis. Which method provides protection to infants against whooping cough and reduces the chance of developing the disease later? Injections of antigens from Bordetella pertussis bacteria. Protection of infants. So you've got to give them some vaccine. So you have to give them the antigens from this bacteria so that they develop the plasma B cells and the memory B cells, and when actually they get the infection, and when actually the bacteria enters, then they will not be getting the infection. Which method provides protection to infants? The other things are all wrong. A short course of antibiotic. Well, no, it's not going to protect them. We're going to give them antibiotics when they get the disease. A 6-month course of one type of antibiotic. Why would you do that? Antibiotics are not very good for you. You should not give them to people indiscriminately without any reason. Injection of antibodies. How do we know they have the infection that we're going to give them antibodies, and you don't make it when we don't have antibodies against these? Question number 38. The electron micrograph shows a type of blood cell. What can be concluded from the electron micrograph? Well, you can see a lot of rough endoplasmic reticulum. That's all what you can see. All this is the rough endoplasmic reticulum. It's a lot of it. So all what it has to do, it has to cell synthesize a large quantity of proteins. Digest pathogenic bacteria. No, for that you need lysosomes. So more Golgi. The cell secretes products that are toxic to pathogens. Well, how can they be toxic to pathogens? But then we haven't studied any such thing which is toxic to pathogens. The cell synthesizes large quantities of antigens. Well, every cell has its own surface antigen. It's not going to suddenly make more antigens. The bacteria has antigens on its surface, or the pathogen has antigens on its surface. So the reason that the rough endoplasmic reticulum means large quantity of proteins are going to be made. Question number 39. Why is mitosis important in immune response? Please remember, mitosis means some sort of cell division and more cells being formed. So it allows B lymphocytes to produce plasma cells. That is the answer to it. But why is B wrong? It allows B lymphocytes to synthesize antibodies. Antibodies are proteins, they're not new cells being formed. Allows neutrophils to carry out phagocytosis. So the neutrophils engulf bacteria, but it's nothing to do with mitosis. Phagocytes are not multiplying and forming more phagocytes. In fact, they are dying. When they destroy, when they engulf and digest and destroy, they die in that process. It allows T lymphocytes to recognize foreign antigens, but that has nothing to do with mitosis and the immune response. Question number 40, last question. What is fused with a B lymphocyte to form a hybridoma cell in monoclonal antibody production? What is fused with a B lymphocyte to form a hybridoma in monocell? So a cancer cell is hybridized, and that is why it is a myeloma cell, which is the D. So what is fused with the B lymphocytes to form a hybridoma? So the B lymphocyte and the cancer cells, when they are fused together, then this is the cancer cell, and then it forms a hybridoma. And of course, these cells become immortal because cancer cells are immortal, and then they can keep on and on producing antibodies for us, which we need. Because what do we want? Monoclonal antibody production. This completes our paper, and I hope this is helpful to you. Please do leave comments, if any, if you want me to do any more papers or anything which helps you in your exams. Thank you very much.